Hello everyone, my name is Jillian Okimoto. I'm 17 years old and I was diagnosed with osteosarcoma in December of 2017. I went through 9 months of chemo and 9 surgeries in a process called distraction osteogenesis, which was able to regrow the gap in my bone. And I will be 5 years cancer-free in August. Yes. So this is my first year at Factor, and it's also my first year with MIB. I joined the Junior Advisory Board this year in January, and it's been wonderful to start giving back to the community that saved my life. A little bit about me. I'm a rising senior and a fine arts major at Frank Sinatra School of the Arts in Astoria. I'm a born and raised New Yorker, which means that I'm a little bit unused to an area where you can't just A, take the subway, or B, walk. And so, I realized that jaywalking in Atlanta is close to impossible. On the way to lunch yesterday, I was almost run over, even though it was my light. So besides art, I'm a baker and I'm a writer, and I love designing video games. If you Google "slide me a cold brew," you should give me a 5-star rating. I'm a younger sister, I'm a foodie, and a proud tea drinker in a coffee-loving household, so it's been a tough life, not just because of the cancer. But enough about me. You all came here for something else. So I'm honored to introduce Dr. Heather Gardner. Dr. Heather Gardner is an assistant research professor at Cummings School of Veterinary Medicine at Tufts University. Dr. Gardner earned her DMV at Washington State University and completed her residency at Veterinary Medical Oncology in Ohio State University before completing her PhD in genetics at Tufts University. Her research efforts center on comparative and translational cancer genomics using the tumor genome to inform novel biomarker-driven therapeutic approaches. Yes, so big round of applause for her.
All right, thank you everyone. And again, thanks for the invitation. I'm really honored to be able to introduce really a great panel of speakers here. And I think it actually is going to follow on nicely from the panel that we had here before lunch talking about preclinical models, because we're really going to be focusing on the comparative oncology side of that preclinical model system. And so first we'll have Dr. Sean Khanna, who is currently with the Osteosarcoma Institute and Ethos Discovery, and a lot of his work is really centered on understanding metastasis and osteosarcoma, and so he'll be talking to us today about scientific translation that is really integral to the comparative oncology efforts. Then we'll hear from Dr. Amy LeBlanc, who's with the NIH and NCI Comparative Oncology Program. She's really done a lot of great work leading veterinary clinical trials and also with a large effort focused on genomics in canine osteosarcoma as a translational model, and we'll hear a bit about that today. And then next we'll be hearing from Dr. Nikki Mason, who is currently at the University of Pennsylvania. She is going to talk to us about really a large effort called Precinct, which a lot of you have probably heard about, but she's going to talk about how that has really impacted veterinary trials with a goal of improving outcome on the human side as well. And then we'll start hearing about some of the projects that are really taking a comparative focus. So we'll then hear from Dr. Dan Reagan, who is at the Colorado State University. He's going to talk to us about some of the work that he's done looking at lung fibroblasts and macrophages in the setting of metastatic osteosarcoma. Next, we'll hear from Dr. Mike Leibovitz, who's at the University of Colorado, and he'll be talking to us about using the dog model to help develop novel therapies in osteosarcoma. And then finally, we'll hear from Dr. John Ligon, who's at the University of Florida, and he's going to be talking about some of the work that was really supported by an Outsmarting Osteosarcoma fund. And we'll be talking about RNA nanoparticle vaccines. So with that, I don't want to take away time from our speakers, so I will hand it over to our first, Dr. Sean Khanna. Thank you.
Thank you very much, Heather. It's a great opportunity to join this great panel, and I'm going to let everybody down, Josh Schiffman in the front here. So there's no beautiful science here. I'm going to share some ideas on this field of comparative oncology and echo Ryan Roberts' comments on scientific translation. We know what doesn't work, And we hope that by knowing what doesn't work, we can improve. And that's part of the iteration that I think is really the punchline to the scientific translation part of the discussion. So I wanted to use a slide before someone else did, and that is, it just briefly says that most of us look more like our pet dogs than we do like mice, and that's true both in the cartoon and it's true in this heat map, which basically shows that you can't distinguish a dog with osteosarcoma from a human with osteosarcoma across thousands and thousands of orthologous genes. And so that leads to the hypothesis that the dog with osteosarcoma can assist in the translation of knowledge from the laboratory to the clinic, or the lab to the Labrador, as some people would say. I think in red, from the standpoint of translation, there are some critical features of the dog that lend itself to some of the points that were discussed earlier, and lend itself to this hypothesis that we can do better by integrating the dog into a translational development path. So every cell in a dog's tumor is different than the cell beside it. Every dog's tumor is different than the dog beside it. And that is the heterogeneity of cancer, which is the essence of the problem of cancer in humans, and it's naturally occurring in dogs with cancer. It drives drug resistance and metastasis, and that's why I think we should consider the dog as a model in this hypothesis. We have, for a long time, and I'm going to take a little bit of the work that Amy's championed and advanced in her time at the NIH and the NCI Comparative Oncology program, imagine that we could do dog studies both because of the lifespan of dogs, the life with osteosarcoma being shorter, and the fact that there are many, many more dogs with osteosarcoma than children. We could actually pick the winner in a series of dog studies with new drugs, and then advance the winner into human clinical trials. So you still have to do human clinical trials. That's the arbiter of what wins and doesn't win. But you can sort of tip the balances a little bit by picking a winner in a series of these dog trials. Am I really 7 minutes in?
You've got 7 minutes left.
Okay, wow. I was thinking, okay, I really have to hurry up. So this was the idea, and the idea was then, as initially imagined, do a study with dogs who present with localized disease, perform surgery to remove their primary tumor, give them a new drug plus standard of care chemotherapy versus chemotherapy alone. But the problem with that is that cycle of innovation is too long to adequately inform what's needed in terms of new ideas into the human. So we somehow have to shrink that cycle of innovation, and I'll talk about an idea we have to do that, but we need to do that within the comparative oncology agenda to make this idea work, I think, more effectively. So most of the work that I've been involved in terms of translation has been with this drug rapamycin, which is an inhibitor of the mTOR pathway, which we believed, for a number of reasons, was important in the process of metastasis. We published this paper, and I'm not going to say much more except the paper said these are all the reasons we failed to effectively translate from the mouse in the laboratory to the human, and you can read the paper. I think most of the failures are the result of us not having adequate knowledge about the biology of the target and the mechanism of action of the drug. We do a little bit of hand waving, and that causes problems. To echo Ryan Roberts' comments and Dr. Healy's discussion, there is no great model. They're all models of a disease, and you can get closer, I think, by doing multiple models, and that includes tissue engineering, mouse models, the dog. And I think a really important point is how drugs are distributed in the body matters when you're talking about developing a drug. So pharmacokinetics and does the drug turn off the target that it intends to, pharmacodynamics is important. So these are the things that I think are critically important, and I believe the dog as a model is a good way to advance this agenda. But the paper is there, and this is a call to the young people in the audience. So from 2014 is when this paper came out. There's been a lot of opportunities for you now to think back in the last 10 years and look at some additional failures and to repeat what we did in that paper and publish a paper that says, how can we do better? So there's an easy paper for somebody out there to iterate on this approach that we described in 2014. So if I talk about the rapamycin, I'm going to jump all over 20 years of research biology that led us to do a dog clinical trial. The dog clinical trial asked the question that I articulated early: does the addition of rapamycin to chemotherapy do better than chemotherapy alone? And you see these overlapping survival curves, so the answer was no, it didn't work. So despite trying to do everything that was in that last paper, we failed, and now this is the start of new iteration. And if you look to the right, you could say what went wrong. So if you look at the little green silhouette, that's when we exposed these dogs to rapamycin in the course of their disease, and that's probably too narrow. And one argument would be, let's do it longer. And to do that longer, we need a better dog formulation of rapamycin. The human formulation of rapamycin is great for humans, but to do the research study, we need a research version of rapamycin to model what is needed to do in the dog, which is a longer exposure of the drug over a longer period of time. So a new formulation is something we're working on to repeat the study in a different way. No single drug has done anything dramatic in any cancer setting, so probably a combination is needed. So we need to explore new combinations. And I have a disclosure related to Vujade and the idea of coming up with new disclosures. So the idea around iterating more quickly and shrinking the cycle of innovation from the dog presenting with a primary tumor, doing a surgical intervention for their primary tumor, and then giving adjuvant chemotherapy until they relapse, is to use innovations in surgery so that we can do what's done in humans, and that's remove metastatic nodules, collect tissue, give them to the biologist to do studies on the metastatic tissue, but then apply new drugs and ask the question, does the drug do something to prevent the second recurrence or the third recurrence in the dog in a way that allows us to innovate in a more rapid manner. So we're doing this with video-assisted thoracoscopic surgery in dogs, and this allows us to take that Great Dane with osteosarcoma and not have to do a median sternotomy and open their chest and have them recover for several days in our ICUs in vet hospitals. This now allows a 40-hour discharge for dogs, which allows more of us to offer this to our patients and do more clinical trials to accelerate the cycle of innovation. So we're doing these studies now, and this would be how we would add a new drug in the future. So I have the disclosure to Vujade. Vujade is a sponsor of this meeting and is dedicated to finding drugs that prevent metastatic progression in osteosarcoma. And through the work that Vujade does, we have hypothesized that we could come up with a second drug to combine so as to advance a combination that would improve the results from that single agent. And if anyone wants to twist David Warshawski's arm, he can give you the details on the winner in that combination, but I'm not allowed to do that, and should make it clear that I do have a financial disclosure relevant to the work of Vujade. So the last phase is, how do we take this interesting hypothesis that's largely a successful academic hypothesis, and how do we sort of gear it up to do this more quickly? So where we are now is we have a national infrastructure to do clinical trials in dogs with cancers. We have clarity on how these studies fit into a regulatory path. We have reagents, we have endorsements from governmental bodies, from biotech companies, and so forth. And we could just say, well, let's just do more of that and get a few more chances at bat, and eventually we'll hit something good. And that would be more of what we're doing. The alternative is summarized in this slide, and that's to add a commercial perspective to this largely academic approach and create a transaction. And this is a figure from a paper, and if anyone wants a reference, you can get it from me. But this figure from the paper basically says that if you could create a transaction, a financial transaction between human biotech and biopharma and animal health biotech and biopharma, that transaction would create a self-funding model to do comparative oncology research and do this in a more rapid manner. One of the things that we've done to advance this agenda, and that's under this tagline, Ask FDA, is to streamline the approval. So when you approve a drug in the human, you have to say you can make the drug effectively, you have to say it's safe, and you have to say it works. And to manufacture a drug shouldn't be different between an animal drug and a human drug. But until very recently, they were different. So we've achieved alignment between the human FDA and the veterinary FDA to say that if you have an approved drug in the human, you know how to make it, it's approved on the manufacturing side in animal health. And that makes the transaction between those two groups now more valuable. Now there's more resources for this group of people to do the research. So these are all the, again, thoughts, no deep science here. I'm happy to sit down and listen to some good science, but really appreciate the opportunity today.
All right, well, thank you for the opportunity to be here. Again, to the organizers, Christina, to my fellow panelists, really, and to the patients and their families. This is just a hugely inspirational moment for, I think, all of us here, and it helps propel us forward into our work. So my name's Amy. I'm a veterinary oncologist and director of Comparative Oncology at the intramural NCI. And my job today is to tell you about our progress in comparative osteosarcoma research under the auspices of what we're calling the Dog Squared Project. So I don't think this slide needs much introduction. I think all of us recognize metastatic progression is associated with a really undesirable outcome for patients, whether they be four-legged or two-legged. And we're lacking in predictive prognostic biomarkers, and we really need to move the treatment and outcome needles because they haven't really moved much in the last 30 years. I think important for those who aren't familiar with management of canine osteosarcoma that our patients generally succumb to metastatic progression and death within 3 years of diagnosis. So the vast majority of them, more than 90%. So we have really only a small group of patients that are long-term survivors. And so this is a major medical problem for the veterinary community. And there's definitely a groundswell of interest in helping improve outcomes for dogs' sake as well. As Chand mentioned, the addition of rapamycin onto standard of care didn't really help us move that needle, but what this particular trial did, though, was provide us with a really unparalleled opportunity that stems from the outcome-linked biobank of samples that was collected during this initiative. So 324 dogs were enrolled on this prospective randomized two-arm trial. Although we have some questions about why the trial was a failure, and I think we have opportunities to look back and sort of interrogate that data further, we have now a really, I think, important opportunity to take those samples and associated metadata and ask some really new, important questions about the biology of the disease moving forward. So we created Dog Squared at the NCI with, again, the support of the Morris Animal Foundation that funded the trial itself, and through a variety of samples that were collected during the trial activities that now total at least 22,000 samples. They span normal tissue, primary tumor tissue, samples that were collected at post-mortem exam. When a subset of dogs passed away, they went to necropsy, and we were able to collect tumor tissue post-treatment. We had multiple aliquots of whole blood, serum, PBMCs, and so again, with those samples in hand and the correlative metadata and the support of the NCI cores for sequencing and characterization, we have this really great opportunity to create, I think, a framework for the disease. Not just a genomic framework, but a pathologic framework, an immunologic framework, and just a deeper understanding of the biology of the disease itself. And all of the primary tumor tissue and its associated normal or tumor-adjacent normal tissue was all collected at the time of limb amputation prior to any chemotherapy. So we have access to this really broad collection of treatment-naive specimens, which is unparalleled and really, I think, an exciting opportunity for multi-omic characterization platforms to be applied. So I'm going to show you some of this unpublished data that was our first foray into transcriptional profiling. So what I'm showing you here is deconvolution analysis of canine transcriptional data. So the first heat map is bulk RNA-seq data from 190-ish, 97, 98 samples. The heat map has, in the yellow, those are primary tumor, but you see also some green denotes some of the metastatic samples that were put in there as well, because we did have metastases collected at necropsy that were subjected to bulk RNA-seq profiling. And so essentially what you do with this transcriptional data set is provide a method for virtual cell sorting that allows you to cluster the tumors into 3 distinct subtypes. So there's an immune-enriched subtype, an immune-depleted or immune desert subtype, and then this sort of intermediate subtype of immune-enriched, but also sort of a stromal vascular enrichment as well. And so we were able to apply that deconvolution technique to the bulk RNA-seq data, but then also some nanostring gene expression profiling using the Canine IO panel that was recently made available a couple of years ago. And so the nanostring data is shown in the other heat map. And so what you see there is 115 samples. The vast majority of them were from metastatic lesions, and this was RNA created from FFPE blocks. But there are some primaries in there as well, denoted in yellow. So I think what you can appreciate from this is that you still see the same 3 subtypes, i.e., IECM and ID, but then there's no correlation between the subtype of the primary and the subtype of the metastases, because we looked and we couldn't find that. So this work's ongoing in terms of being able to apply IHC to say, well, if the deconvolution technique says these cell types are there, you really sort of want to see them in tissue to make sure that, yes, if this immune-enriched subtype is there, well, you sort of want to see the cells that are theoretically responsible for those transcripts. So that work's ongoing. So after we discovered this, we thought, well, let's look more deeply in terms of functional genomic features. And so this is just pathway GSEA analyses of the different subtypes that show, indeed, there are distinct pathway alterations that can be attributed to each of these subtypes. And then taking it a step further, the clinical outcomes of these patients are shaped by these TME composition subtypes. So certainly, as you might expect, the immune-enriched cases, and this is all canine, do much better with equivalent therapy than their immune-depleted or immune-desert counterparts. And when you look at relative abundance of cell types, you appreciate that the patients, canine patients that had a longer survival had enrichment of T-cell subsets, as opposed to the poorer outcomes had more enrichment of endothelial and ECM stromal components. So the follow-up to this is obviously some spatial profiling to understand the relationships between these cell types and how they refer to each other geospatially, but also within the compartment of the bone itself and other anatomical barriers to, say, immune cell infiltration. So we take a machine learning analysis to apply this same approach, and it turns out that these TME subtypes are also predictive of outcome in people. So they're present in both canine and human, and then they actually also predict outcome in people. So here I'm showing you the same algorithmic approach, an adversarial model that plots, pits really the dog versus the human computationally against each other to create then the designation of immune enriched, immune desert, and immune enriched ECM subtypes. And so this was done on publicly available data from Target and 3 other publicly available transcriptional data sets, from which we had clinical outcome data from 160 patients. And so what you're seeing here is that the algorithm that was detected in dogs and developed in dogs applies directly to the human transcriptional landscape. And in 53 of these patients that were non-metastatic at diagnosis, and in that sense, they really match what dogs typically, how they present to us clinically. They don't have macroscopic metastatic disease usually in their lungs, but we know it's there at a microscopic level. If you take 53 of those sort of same types of human patients, this approach of this 3-compartment designation predicts metastases within 5 years of diagnosis. And then just one other sort of aspect of this, that looking at macrophages is one thing that we're looking at in some more detail. And so M0 macrophages appear to be more associated with a poorer outcome in both dogs and people, whereas an activated M2 appears to be enriched in those that had a better outcome. And then when we do some Spearman correlation with this unsupervised clustering of that CyberSort virtual cell sorting data, again, gives you just a little bit more insight into the subtypes that segregate by prognostic outcome, so favorable versus poor. So again, in the dog, you see a correlation amongst those activated T cell populations. It's a little bit different in people that have a favorable outcome, but the theme is the same in that the more immune enriched your tumor is, the better off the outcome would be. We have also the opportunity, again, in that setting of necropsy data to look at dogs that went through a postmortem examination, and we've identified a cluster of dogs that did not develop lung metastases. Really, they got bone mets and not really a whole lot of mets overall, but they were still subjected to the same type of therapy and a thorough postmortem exam. They didn't really have any differences in their outcomes in terms of survival, and that's again reflective of veterinary oncology and veterinary medicine in general, where survival in dogs is really more a reflection of their owner's wishes rather than their disease status in some cases. So we're taking a closer look at this cluster of dogs that for some reason was not as apt to develop lung metastases, and wondering sort of why that might be. So more data will be coming and on that theme in the future. But I think what I want to leave you with is that these types of clinical trials are an incredibly valuable resource that enable discovery and validation of predictive biomarkers that have value in both species. And we're continuing to work on complementing the transcriptional data set with whole genome and whole exome methylation profiling, and the first comparative osteosarcoma pathology board as well, which is featured here and has the goal of really getting people together to talk about the histopathologic and immune and other histologic features of osteosarcoma in both species, and really start talking in more detail about how those data sets can be layered on one another to create a more informed method of diagnostic classification. I know I'm over, but I'll leave you with this, that this is the current landscape of COTC clinical trials that we run from our program at the NCI. You'll see a lot of them have to do with osteosarcoma. We recently got a collaborative R01, so it's a U01 award with Dr. Rob Rabune at the University of California, Davis to do a natural history study in osteosarcoma. So that will be, I think, the next iteration of data that we'll be able to collect from dogs with treatment-naive disease, and think more carefully about the types of biospecimens we want to collect. And it will open up additional opportunities, I think, for single cell, single nuclei sequencing, PDX generation, et cetera. So I'll thank all the folks that helped. Chan, of course, none of this would be possible without you. So thank you so much for laying the groundwork for our community, and look forward to your questions.
All right, thank you very much. First of all, I'd like to thank Christina and Anne and the whole of the MIB family for the invitation here. I always find this meeting incredibly inspirational, and it is just a pleasure to be here. My job today is to introduce you to Precinct, and that is the pre-medical cancer immunotherapy network for canine trials. So this network is a group of individuals with a diverse set of expertise and interests in comparative oncology and comparative immuno-oncology. And by building this network and bringing this group of people together, we hope to use innovation and collaboration and data sharing to accelerate scientific discovery and bring new therapies into the human clinics. So how did this come about? Well, it was born out of the Cancer Moonshot Initiative, which has already been mentioned this morning, which came about in 2016. And the overarching goal of that initiative was to essentially compress 10 years of discovery in cancer into 5. So how could we actually accelerate scientific discovery? And ideas that came from that were, well, we need to foster greater collaboration, we need to improve sharing of cancer data amongst the research community. And in fact, as part of this Cancer Moonshot initiative, a blue ribbon panel was put together, and recommendations came out of that panel in order to achieve this sort of goal. And one of those recommendations was to build these translational networks to bring people together, to encourage collaboration. And Precinct was one of those networks. And so again, this is a network that focuses on immunotherapy of cancer using immunocompetent pet dogs with spontaneous tumors to accelerate information and to inform human clinical trial design. And so the idea of Precinct was really to sort of facilitate performance of these immunotherapy studies that were being performed by investigators that had received UO1 awards to trial immunotherapies and combination immunotherapies in dogs with cancer, to support correlative studies that would identify biomarkers of immunological response and also of therapeutic response, and essentially to catalyze research productivity. And so the first 5 UO1 awards are shown here, and it's sort of, I feel it's like a who's who of key opinion leaders in comparative oncology, and you can see the institutes that were awarded those, and I'm going to tell you a little bit about some of those studies in a minute. And so Precinct obviously consists of these individuals. There's a data coordinating center within Precinct that provides support where needed for biostatistics and bioinformatics for those trials. And also the network integrates with the immuno-oncology translational network and also the pediatric immunotherapy discovery and development network as well. And actually born out of this sort of data coordinating center, we've worked very closely with another initiative that I'll talk about at NCI, which is development of the integrated canine data commons, which has an increasing amount of data in there that can be very readily mined, and we'll talk about that in a minute. So this is just an example of the cancers that were being evaluated using various different immunotherapies and combination immunotherapies. So we had 2 of sites, University of Minnesota and the University of Alabama, that were working on immunotherapies for the treatment of high-grade glioma. That included at Minnesota a tumor lysate, an autologous tumor lysate vaccine, and a checkpoint inhibitor peptide, and then down at UAB, oncolytic virus therapy. Then we had Cheryl London's group at Tufts working in lymphoma with anti-CD20 monoclonal antibody in combination with small molecule inhibitors. And then we had 2 groups working in the osteosarcoma field and also metastatic osteosarcoma and metastatic melanoma at UC Davis. They were looking at IL-15 and a superagonist IL-15 in combination with autologous NK cell therapy to treat dogs with pre-existing metastatic disease, and then moving forward into primary disease as well. And then the group at CSU repurposing drugs, including losartan, and combining that with other TKI, basically, to try to alter the metastatic niche in the lung. And I think what's really important when you look at these 5 studies, 2 of these studies have supported clinical trials going into human clinical trials. So the tumor lysate vaccine and checkpoint peptide inhibitor at Minnesota is now currently in a clinical trial in humans for recurrent glioblastoma, and the work from Steve Dow and Dan Reagan is actually in a phase 1/1b clinical study now in relapsed refractory osteosarcoma. So I think this is really a win to see that data from the dogs is helping to inform those clinical studies. We've had a fair amount of publicity supporting this concept of comparative oncology, and this is just an example of some of the manuscripts that have come out of this work, and that is in the scientific community. That's again a win. We're able to spread the knowledge that we've produced from this initiative. So there is a second round of UO1 awards, actually 2022, this is a typo, I apologize, for the same thing, comparative oncology and immuno-oncology. and bringing together collaborators. And you can see now we're starting to see groups coming together where we're seeing veterinary clinical scientists collaborating with the human scientists as well. And so we have quite a focus this year in osteosarcoma with 2 groups working in that region. Again, Cheryl and Gillian Richmond working with tumor vaccines, chemotherapies, and small molecule inhibitors, those combinations in osteosarcoma. And then we have a cellular therapy that Dr. Rotolo will talk about, I think, tomorrow, using redirected iNK T-cell platform for osteosarcoma. We have 2 groups focusing in urothelial carcinoma with cytokine therapies and checkpoint inhibitor antibodies, anti-PD-L1 with Debbie Knapp. And then we also have Jaime Modiano and Shruti Naik at University of Minnesota and the Mayo Clinic looking at oncolytic viruses and checkpoint inhibition, again in lymphoma. So again, I think the fact that we've got another round of funding from NCI is also, again, encouraging. Part of Precinct also has been to establish the central data management system. So our data coordinating center will develop databases, clinical databases for groups that don't have them at the UO1 sites. The clinical research coordinating unit at Penn, part of the DCC, is involved in curating that data and basically then putting it into the appropriate format to upload into the integrated Canine Data Commons that can then be easily shared with other users. I'm encouraging people that are interested in a comparative approach to go to the ICDC website, which is shown here. There's about 35 terabytes of data in that data commons currently. And the data that you can download and mine is quite varied. And I'm sorry, this is very small, but basically you can download the study protocols that are being used in the clinical trials that are here. You can look at clinical data, clinical pathological data. You can look at genome sequencing of the tumors, RNA sequencing, cytokine data, and so forth. So I really encourage you to go there and start to have a look at that. We've got 6 or 7 other studies that are going to be uploaded over the next couple of months as well. And I think that the idea is to encourage people to take a look at this and see if the dog is going to actually serve potentially your purposes and can help you advance your science. Also part of Precinct, we've been interested in immunological tool development and reagent development. We need to be able to deeply interrogate immunological responses of our canine patients so that we can really understand how these types of immunotherapies are working and provide an appropriate parallel for the humans as well. Amy spoke of the Canine IO panel, which members of Precinct worked closely with NanoString to develop, and this enables us to evaluate 800 genes, mostly immune-related, to again interrogate responses in dogs that have received these various immunotherapies. We also can make antibodies as well. So if we have interest in looking, for example, at B cells in the tumor and peripherally, we can do that, and we can develop these antibodies and validate those appropriately. And then also our group has got together and looked at particular flow panels and gating strategies that if you want to, you can utilize those. The idea is for us all to be able to harmonize the data that we're getting from these sort of disparate clinical trials and be able to leverage it further than it would if we were all using different panels or different gene expression platforms and so forth. And so I just want to sort of acknowledge the people. This is sort of the 2017, the old guard, not so old, sorry, the young old guard, who basically have sort of poured their energy and spirit into this initiative and have made it really a solid, I think, network for collaboration and innovation. If you are interested in joining Precinct, we have monthly steering committee meetings and presentations there. Please just shoot me an email. I'm happy to add you to the list. So with that, thank you very much for your attention.
Okay. Thank you, Heather, for the introduction, and thank you, Christina, and the organizing committee for bringing me back this year. As everyone said, this is a very inspirational and motivating opportunity to be here. So I'm going to talk a little bit about some of the work that led to our Outsmarting Osteosarcoma grant and then work we've been doing over the last year. So The overall goal of the lab is to leverage spontaneous osteosarcoma in dogs, among other cell culture models, rodent models we have in the lab, to understand the cell types and the molecular mechanisms that are really shaping the evolution of the tumor microenvironment, specifically within the context of lung metastases. And to do that, we really are taking a multi-pronged approach, and we're focusing primarily on 3 cell types that I'll tell you about today that we think really have a significant role in promoting metastatic progression in the lung. So the first cell type we're talking about is really some work we published recently, which was really standing on the shoulders of Jeffrey Pollard and his work showing an important role for CCR2 expressing inflammatory monocytes in promoting lung colonization and metastatic outgrowth in other tumor types, primarily in breast cancer. And so we showed that in osteosarcoma, both osteosarcoma cells and other stromal cells within the lung metastatic site overproduce the chemokine CCL2 that can recruit this same population of CCR2 expressing monocytes into the metastatic niche to provide tumor-associated macrophages for these metastases. And we went on to show that the repurposed CCR2, the repurposed antihypertensive drug losartan could be repurposed as a CCR2 antagonist to block this CCL2-mediated monocyte recruitment. And we combined this drug with the multi-kinase inhibitor tosiranib, which is functionally equivalent to the human drug sunitinib. We saw significant clinical benefit in a clinical trial in dogs with lung metastatic osteosarcoma. And ultimately, this data, as Nikki mentioned, led to a human phase 1 clinical trial that's currently ongoing. But in the last few years, we've really shifted our focus to looking at a population of fibroblasts within lung metastases and specifically in dogs. And this is work that's being led by Eric Palmer, a PhD student in my lab. And this interest in lung fibroblasts was really driven by an observation where we observed activated focal adhesion kinase signaling within osteosarcoma, both canine and human. And so, as I'm sure many of you are aware, FAK is a non-receptor tyrosine kinase, and it's been shown in a number of other tumor types to drive numerous cell-intrinsic pro-tumorigenic functions. So it can promote cell survival, proliferation, migration, stemness, and we've observed it to be hyperactivated both in canine and human osteosarcoma tumor cell lines and metastases. But what we were interested in is specifically its role in perhaps shaping the microenvironment. So we observed that we could target this activated FAK signaling with a FAK inhibitor, defactinib, and we could significantly inhibit cell proliferation and survival and extend survival in mouse osteosarcoma lung metastases models. But this molecule had also been shown to drive chemokine gene expression, both in the context of the stroma of pancreatic cancer, as well as within squamous cell carcinomas in mouse models and human patient samples. And so we sought to explore whether FAK signaling within specifically osteosarcoma could really shape the composition of this tumor microenvironment. And so using bulk RNA-seq data for both canine and human tumors and stratifying these tumors as either FAK activated or FAK high versus FAK low, we saw significant enrichment of immune gene signatures associated with immune suppression within the tumor microenvironment. So as you can see pointed out here, we have enrichment for pro-fibrotic and pro-metastatic cytokine and chemokine expression, monocyte and macrophage recruitment and differentiation, and T and B cell suppression in these FAK-activated tumors. And this data has also been supported by a single-cell RNA-seq analysis of canine osteosarcoma lung metastases, where not only did we observe FAK expression both within the tumor compartment, as I've already showed you, but again, specifically within these lung fibroblast populations. Here, they have significant overexpression of FAK, and importantly, these fibroblasts are one of the top producers of the monocyte chemokine and the losartan drug target CCL2. And again, using primary human donor-derived lung fibroblasts, we were also able to confirm this FAK hyperactivation within this lung fibroblast compartment. And so using a variety of in vitro assays where we looked at the role of osteosarcoma-derived cues in activating this lung fibroblast population, and namely exosomes, which you heard about earlier this morning, we showed that treatment with this FAK inhibitor, defactinib, was able to block osteosarcoma exosome-induced lung fibroblast proliferation, as well as secretion of that monocyte chemokine CCL2. And so the reason we feel this effect of this FAK inhibition on these fibroblasts is important is, not only for the role of their ability to block this potential important cell source of CCL2 and its role in recruiting macrophages to these lung metastases, but also in using these in vitro co-culture models, we've shown that these exosome-activated lung fibroblasts provide a unique survival advantage to these osteosarcoma cells under conditions of serum starvation or cell stress. So as you can see here, these osteosarcoma cells in monoculture alone in green are unable to survive in culture over the course of roughly 2 weeks. But when you co-culture these cells with these primary human donor-derived exosome-activated fibroblasts, you see significant increases in survival in these cells, and that's shown here in red with live cell imaging. And so ultimately, this data led us to ask whether adding this FAK inhibitor through its ability to have combined targeting of both the osteosarcoma cells as well as these lung fibroblasts could improve the clinical benefit rate of our macrophage-targeted immunotherapy, which is losartan and tesiranib. And so far, we've performed a FAK-inhib pharmacokinetic analysis in healthy dogs where we've observed very favorable exposure for this drug. So we're meeting or exceeding recommended phase 2 doses for defactinib in human patients, and we're able to get significant target modulation in the peripheral blood of these dogs and inhibition of phosphorylation of FAK in this study. And so far we've recruited 6 of 12 planned canine patients with lung metastatic osteosarcoma into this trial, and hopefully have some updates for you next year on that data. So lastly, I just want to shift gears to the third cell type I'd like to talk about, which is the alveolar macrophage. And a question we're interested in is whether these cells could perhaps serve as a sentinel of this evolving or changing lung metastatic tumor microenvironment in osteosarcoma. So really, some of the rationale for investigating this cell type is that in our mouse models, we've observed that these osteosarcoma exosomes, that alveolar macrophages are one of the primary, CD11C positive alveolar macrophages are one of the primary cellular targets of these exosomes. So when these exosomes distribute into the lung, they're taken up by these macrophages, which we can detect. And importantly, they're also eliciting this chemokine secretion that I've showed you, and namely elevated levels of CCL2, which are uniquely detectable in the airway fluid or this BAL fluid, but not necessarily in the plasma of these mice. And so we're interested in kind of using this rather long-standing diagnostic technique of other lung diseases, which is bronchoalveolar lavage, to determine whether this could be perhaps a new immune monitoring approach for dogs, for clinical trials in dogs with metastatic osteosarcoma. And so to kind of demonstrate our ability to leverage this approach, we've conducted a pilot study where we've looked at 3 healthy dogs and 5 dogs with localized appendicular osteosarcoma, and then 5 dogs with lung metastatic osteosarcoma. And we've performed single-cell RNA-seq analysis of BAL fluid from these dogs. And what we've observed is we can find 40 different cell populations in this BAL fluid, including 16 different subsets of macrophages. And 11 of these 16 populations of macrophages are enriched in dogs with localized primary osteosarcoma as compared to our healthy controls. And importantly, the abundances of some of these clusters of monocytes and macrophages is positively correlated with FAK expression, and is also correlated with CCL2 levels as measured by ELISA, uniquely in the BAL fluid, but not necessarily in the plasma of these dogs. And so again, our goal is to kind of leverage this technique in monitoring both baseline and post-treatment responses in these patients to see if it could perhaps be more informative than peripheral blood monitoring in these dogs. So in summary, really, our goal is to identify these cell-intrinsic regulators of the osteosarcoma tumor microenvironment composition, identify combination therapies that target both of these compartments in the tumor, both the stroma and the tumor cells themselves, and then identify correlates of response to these therapies. So with that, a lot of thank yous here, and again, thank you to the organizing committee for the opportunity to be here today.
Hello. I am Mike Leibowitz, and I think I am the first pediatric doctor. Okay. All right. Sorry about that. So I want to talk a little bit about a collaboration that started between Dr. Regan and I at Factor last year. Even though we're both from Colorado, we didn't know each other, and we met at Factor and liked each other's ideas, and we decided to team up and collaborate. All right, so I'm going to talk a little bit about FAP, not FAC. FAP is fibroblast activation protein. It's a serine protease. It's been associated with tumor progression, metastases, and poor prognosis when expressed in a number of solid tumors, including osteosarcoma. FAP is not only expressed in osteosarcoma tumor cells, but also by another cell type called cancer-associated fibroblasts, which are recruited by these tumors. And these cancer-associated fibroblasts are quite immunosuppressive. So they secrete immunosuppressive cytokines, chemokines. They express molecules that down-modulate immune responses. So our therapeutic target of FAP not only targets the tumor cell, but these fibroblasts as well. What about on-target off-tumor effects? So FAP is mainly expressed during embryonic development, as well as in fibrotic states. So idiopathic pulmonary fibrosis, cirrhosis of the liver, things that you don't really see much in children, so we don't expect a lot of toxicity associated with that. In addition, FAP is generally not expressed in benign tumors, and quiescent tissues. So the immunohistochemistry here, let's see if I have a pointer. Okay. The immunohistochemistry here, so this is just from another paper in China showing a significant amount of FAP expression. Back when I was at Penn, we had tumor microarrays, and I did FAP IHC in Ellen Pure's lab, and we found FAP expression in osteosarcoma. In addition, when you look at the human cancer cell line database, we see much more FAP expression in osteosarcoma compared to other solid tumor types. I also looked at other pediatric solid tumors, including Ewing sarcoma and hepatoblastoma. So more desmoplastic tumors tend to have FAP expression. Okay, so this was work that was done by Ellen Pure and Steve Albelda at Penn. And previously, they developed a mouse FAP CAR. Just walk over here. A mouse FAP CAR. And this is an in vitro assay. Whoops. I'm sorry, guys. Where they injected FAP CAR T cells into mice bearing this TC1 tumor cell line. And you can see that these FAP CAR T cells were able to inhibit tumor growth, but ultimately these tumors grew out. So FAP CAR T cells as a monotherapy doesn't seem to be efficacious. Here is second harmonic generation microscopy. I mentioned that FAP CAR T cells don't only target tumor cells, but they target these cancer-associated fibroblasts. So what you're seeing in green here is fibrillary collagen, and these fibroblasts basically secrete collagen and extracellular matrix. And you can see that the FAP CAR T cells were able to decrease the amount of fibrillary collagen. And the thought is, by removing that barrier, if you will, more immune cells will be able to invade and attack the cancer. All right, so finally I'm going to get to this canine osteosarcoma project. Again, this came about from chatting with Dr. Regan last year and then after Factor. I think all of the vets know that canine osteosarcoma has a much higher incidence in large breed dogs compared to humans, so it seems like a good model. They develop osteosarcoma spontaneously, and they also metastasize to the lung like in humans. And a lot of the driver mutations like P53 is also shared in the canine model. So I don't need to mention this too much, but Dr. Regan just mentioned he had published a paper showing repurposing of an ARB, losartan, in combination with toceranib, was able to decrease pulmonary metastases. And Dan showed that data last year, and with the FAP CAR that I have at my disposal, we decided to join forces and see whether our combination therapy may be more efficacious than either alone. Oh, and I should mention, Allie McMillan's a postdoc in my lab that's taking over, or taking on this project. So the first thing we wanted to do is determine whether Canine fibroblasts express FAP, like what we see in the human. So this was work done from Dan's lab, where he took a metastatic osteosarcoma lesion. I'm pointing here like you guys can see this. I apologize. And doing a Western blot, you can see that this osteosarcoma expresses, this metastatic lesion expresses FAP. In addition, when looking at FAP expression and how it correlates with CCL2, and as you recall from Dan's talk, the CCR2, I believe, axis is what recruits macrophages or contributes to recruiting macrophages into the TME. You can see there's a positive correlation there. And this is an immunohistochemistry of a canine lung osteosarcoma, where you can see there's a significant amount of these spindly cells or cancer-associated fibroblasts. So it appears that the dog and the human both have CAFs in their lesions. In addition, I believe a postdoc in Dan's lab was able to do single-cell RNA seq, and they identified 4 distinct populations of fibroblasts and found that the vast majority of these fibroblasts express FAP. Dan just showed this picture, so I'll skip it, but essentially it's correlating CCL2 secretion with the fibroblast population. So it would be a good target for our CAR T cells. We wanted to look at FAP expression in the tumor cells themselves. So at CSU, Colorado State, at the vet school, they've developed just a huge plethora of canine tumor cell lines. And when they looked at FAP expression, or at least at the RNA level, it's much higher compared to other histiotypes, other solid tumors. In addition, when they actually looked at the protein level for FAP expression, I believe these are the dog names, you can see there's a variety of FAP expression. Not all of them have high expression, some have lower expression. And when looking at human solid tumors, you can see that bone cancers such as osteosarcoma have the highest RNA level of FAP expression. And here is a lung metastatic osteosarcoma, and these are the tumor cells, and you can see these tumor cells express FAP as well. So I've never made canine CAR T cells. I've only made mouse and human. With the help of Nikki Mason, who's right over here, who has a beautifully published paper on a protocol to generate canine CAR T cells, we're able to generate second-generation CAR targeting FAP. Again, I was going to point down here. We're able to get about a 30% transduction efficiency, which isn't horrible. When we did a killing assay using canine osteosarcoma tumor cells, this is an excelligence assay, and you can see at the higher E to T ratio, 10:1 effector to target, we're able to completely abrogate tumor cell proliferation. At a lower E to T ratio, the tumor cells proliferated and then ultimately were eradicated. In addition, we wanted to make sure that our CAR T cells were activated. So we assayed the supernatants from our killing assay, and just like in human and mouse, they secreted interferon gamma, which is a measure of T cell activation. So we were happy with this. We were able to generate canine CAR T cells. They were able to kill, and they were activated just like we see in mouse and human CAR T cells. So this led to perhaps an overly ambitious grant proposal between Dan and I from the V Foundation, where they had some money to support pediatric cancer research. So thankfully it was funded, and what we're gonna do initially is do a dose finding study. So we're gonna generate FAP CAR T cells, and dog owners with dogs that have osteo are going to decide whether they want to enroll in the clinical trial, and we're going to do a dose-finding study where we inject different doses of our FAP CAR T cells. We're going to look for adverse events, severe adverse events, and determine the appropriate dose of CAR T cell to infuse. After we determine that dose level, really, the aim is to include dogs with both local and metastatic osteo And we're going to use a combinatorial therapy using Dan's losartan and tosarenib with CAR T cells. We're going to give lymphodepleting chemotherapy just like we do in people. The neat thing about this is we have about 2 weeks, maybe 3 weeks, to hold off on biopsy of the tumors in dogs. So we plan to administer these interventions, and then 2 to 3 weeks later, when the biopsy is performed, we can look at changes in the tumor microenvironment. And there's a number of correlative studies we're really interested in looking at, like interrogating the TME and also systemic changes within the blood. So pretty exciting stuff. We hope to open this trial probably by the end of the summer, early fall. There's IACUC, IRB, and so forth that we need to get through. So with that, science is a team game. So I want to thank all of my collaborators. So Dr. Regan, Dan, and again, I'm pointing there, has done a lot of work to make this collaboration possible. In addition, there are medical veterinarian oncologists that are participating. At CU, Mike Verneris is my boss and a great mentor. And a lot of this work really bore out from work that Ellen Pure and Steve Albelda had done at Penn, and John Maris was a mentor at CHOP. So, and of course, I want to thank all the funding we have. Happy to take questions later. Thank you.
All right, thank you all again for inviting me here. And so my name is John Ligon. I'm an assistant professor at the University of Florida. And I'm here, I'm going to give you some updates on our project that was funded last year by MIB agents on our Comparative Oncology program using RNA nanoparticle vaccines against canine osteosarcoma. And then I'm also going to talk a little bit about how this really has helped lead directly to our clinical translational efforts on bringing this into human patients. So these are my disclosures. The main piece is that I'm discussing an investigational agent that is in phase 1 clinical trials. So to give a little bit of background on our RNA nanoparticle vaccine platform, it's really a tumor agnostic approach. So you start with the patient's tumor. In this case, on the slide, I'm showing you an osteosarcoma. You take all of the RNA out of the tumor and attach it to this custom lipid nanoparticle carrier, which you then administer intravenously with the idea that you're going to have really 2 effects. One, you're going to have a localized effect, which leads to turning cold tumors like osteosarcomas into a more hot tumor, meaning more inflamed, more immune active. And then the second part is that because you're giving it intravenously, we believe that this leads to systemic activation of all of your immune organs. And really the eventual idea is that the nanoparticles are transfecting your antigen-presenting cells or dendritic cells, which are then able to present tumor-associated antigens to your T cells and lead to anti-tumor activity. So really, this is a very personalized approach that is unique to each tumor and unique to each patient, and you just know that at the end, you're targeting antigens that are unique to that patient. And the reason that we became interested in using this for osteosarcoma was that by using mouse models of K7M2 osteosarcoma, we really saw that this impacted the tumor microenvironment of murine osteosarcoma. At the top, you have these tumor-associated macrophages and myeloid-derived suppressor cells, and these are bad immune cells that are immunosuppressive and prevent the immune system from killing cancer. And the mice that received the total tumor RNA nanoparticle had complete depletion of these immunosuppressive cells, while on the bottom you have activation of your dendritic cells and T cells. So you really have a complete reprogramming. You're getting rid of the inhibitory myeloid cells. You're having activation of your adaptive immunity as well. And we're really excited about this platform, which has already entered phase 1 clinical trials for adults with glioblastoma. And just to give you a little bit of background on the kinds of results that we're seeing in terms of biological activity, I have 3 rows here. On the top, you have cytokines, which are kind of inflammatory mediators. So anyone who's used a CAR T cell has heard about cytokine release syndrome, and it kind of leads to fevers and chills and other systemic effects. Then you have chemokines, which leads to the movement of immune cells within the body. And then finally, at the very bottom, we have just regular blood counts. And I referenced CAR T cells because I think it's useful to compare, with CAR T cells, you'll usually see cytokine release syndrome with a CD19 CAR T cell somewhere between like 24 hours and like maybe 4 to 5 days after giving CAR T cells. I'm showing on this graph actually levels of cytokines at 2 and 6 hours after getting the vaccine. You're seeing these spikes in cytokines such as TNF alpha, interferon gamma. I think not on this slide is actually IL-6, but this is another very important cytokine that we're seeing. Similarly, you're seeing spikes in these chemokines. All of these that you see, CXCL1, CCL2, and a number of other chemokines, potentially leading to the migration and margination of these immune cells throughout your body. And then I think in some ways the most kind of remarkable number on here is the most simple, which is the complete blood count. I'm showing you here on the left side is the absolute monocyte count drops by more than half after just 2 hours after receiving the vaccine. And then in the middle, you have the absolute lymphocyte count, which drops by more than half at 6 hours after receiving the vaccine. So, what we believe is happening is that as we're giving these intravenously, this is leading to activation of the immune system, margination of your monocytes from the peripheral blood into lymph nodes, into the tumor microenvironment, which are then similarly drawing your T cells out of circulation after that, and leading to education of your immune system against the tumor. So with all of that, I now want to shift gears and talk a little bit about our comparative oncology model that we've been using at UF, and really with some outstanding collaborators, including Rowan Milner, who's you're going to hear from tomorrow, Sheila Carrera-Justiz, and a number of other people who the work that we've done with them has led to this agent entering clinical trials, and I think will lead to us entering clinical trials for osteosarcoma soon as well. So last year, based on kind of the background that I gave you, we discussed that we wanted to determine the safety and the efficacy of RNA nanoparticles in the canine osteosarcoma model. And then we also wanted to look at how the RNA nanoparticles alter the immune architecture, both within the tumor and in circulating immune cells. And we also had an exploratory intent to combine our RNA nanoparticles with radiation, based on one canine with glioma, actually, who had a remarkable response after receiving RNA nanoparticles plus radiation, really a complete response after seeing this combination. So this was really our hope, was to also be able to combine these two treatment modalities. So what I can say is we did have 3 pet dogs that were able to be enrolled this year. We had 10 overall that were screened. One dog that I'm going to talk about was able to receive 4 vaccines, and then we had 1 pet dog which received 1 vaccine and then unfortunately died from progressive disease. And I think that this, speaks a little bit to the difficulty of this model, which I think has been alluded to. We really wanted to target dogs that have metastatic disease at presentation, because that kind of mimics the relapsed osteosarcoma setting. And I think it really speaks to just how quickly canine osteosarcoma can progress, in some ways even more quickly than human patients. So this is something that we're going to try and be more aggressive on recruiting the patients quickly, and possibly combining with other agents such as, again, radiation or an immune checkpoint inhibitor. But this has been a struggle this year. And then another struggle of this model was that we had 1 pet dog who was enrolled but wasn't able to be treated because they were overly aggressive. This is it takes a whole day, honestly. So like the dog has to have the IV catheter in for a full 12 hours. They need to be monitored, in an inpatient veterinary hospital setting, and so it's been a real learning curve, something that I think Rowan appreciated more than I did when we started with this. But regardless, I do want to highlight the bottom line, which was that when you include these dogs that enrolled and the dogs that had been treated before the start of our grant, we now have 5 pet dogs with osteosarcoma. who have been treated without significant adverse events. And so ultimately, this is a big piece of what we wanted to show, is that this was safe in dogs. And that is a huge win for being able to move this into human patients. Now, I do want to talk a little bit about our canine who received 4 vaccines. And again, I'm not sure that this is going to come out very well for those of you in the back, but this was a dog who enrolled with metastatic disease in the lungs. And so this is pre-vaccine, this is after 3 vaccines, and then this is after 4 vaccines. And you can see after the 3rd vaccine, I'm not sure if you can see it, but there are a couple of nodules here at diagnosis that are definitely smaller after 3 vaccines. And then after the 4th vaccine, I actually cannot identify any of these small nodules in the lungs. So this is really kind of remarkable to see an objective response of these osteosarcoma pulmonary metastases. And really exciting for potentially moving this into a human clinical trial. Now, I do have to say, and so you guys cannot accuse me of only showing my best data, we did then also have this radiograph at the same time. So we did notice that they had a large new mediastinal mass. Now I'm showing you the date here. It's just earlier this month. I actually don't know what is going on with this dog, but there's a lot that we need to learn and understand. I do still believe that it's really remarkable for us to see this objective response in the pulmonary nodules, though. So again, these are very clear lungs, and I think that this, and for what it's worth, the dog was very well appearing when we saw them in clinic that day. So a lot more to come, but I think this is the reality of what we're doing. We're actively learning from these patients. And then in terms of learning about biological activity, I think the most important thing I can show you here is I'm recapitulating the blood counts that we were seeing from our human patients. So at 2 hours, your monocytes are going down by more than half. At 6 hours, your lymphocyte counts are going down by more than half. So I think this really speaks again to the fidelity of the canine model when you're comparing to the activity that we're seeing in human patients with glioblastoma. And then I'm running out of time here, but one of our patients, we were able to perform a necropsy, and we're able to get all of this tissue after the patient had been treated with our RNA nanoparticle vaccine. And we are actively analyzing these samples, and I believe that Rowan actually will probably share some of that data tomorrow. Finally, I did mention that we're going to try and combine this with radiation, and I think that this will be an important potential combination as we try to move this into the human clinical trials as well. And I am running out of time here, but I do want to just really highlight that as a result of this comparative oncology model, this has really been foundational in our application to getting an expansion of our IND, which was originally to treat patients with glioblastoma. We now have been approved to treat patients with osteosarcoma as well. And this is just the general schema of our human clinical trial, which will be opening through the National Pediatric Cancer Foundation, hopefully by the end of this year. And many thanks to all of the people at NPCF who have been helping us to put together this project, including several people who are here in this audience today. With that, I will go ahead and just thank everyone who has contributed to this project. Again, particularly my mentors, Elias Sayer and Dwayne Mitchell, who have been incredibly important in mentoring me, Rowan Milner, who's been an outstanding collaborator at the vet school, all of the members of both my lab and the people in Elias's lab, and all of the people who have been supporting us at NPCF, and all of our funding agencies, including MIB. So thank you all very much, and I'm looking forward to any questions.
All right, so thank you to all of our speakers again for a great session. So there's actually a few good questions that popped up in Slido, so I thought maybe we'd start there. So we've been talking obviously a lot about osteosarcoma in dogs as a model, but for those of us who maybe don't think about osteosarcoma in dogs, can someone maybe speak to, how often do we see this disease in dogs? How often is it treated? And just maybe give us a bit more background on what this looks like in our canine patients. I don't know if Amy, maybe you want to take that one?
Yeah. So patients are found by their owners seeking care usually through their community family practice veterinarian. And there's a decision made about whether further treatment versus palliative or humane euthanasia will occur at that point. There's a percentage of pet owners who will choose to be referred to a secondary or even a tertiary referral center where there's oncology expertise and surgical expertise for more definitive management, or exploration of higher level palliation or entry into a clinical trial. I actually don't know the percentage of overall dogs that are diagnosed with osteosarcoma as a percent of the total, but I know that there are people working on developing a canine cancer registry for that very purpose, to get a better idea of the denominator for these kinds of questions. And I don't know the answer to what percent get treatment. There's certainly a lot of pet owners who can't afford or choose not to treat their pet definitively. And that's within their right, of course. And I think providing palliative pain management and end-of-life care is important, no matter what their decisions are. So I don't know if others on the panel have comments for that. Yeah, go ahead, Sean.
Yeah, just to quickly add that we've studied across all cancers, not just osteosarcoma, and on the coasts, it's about 50% of cancer diagnosis get a referral to a veterinary oncologist, and most of those who then see a veterinary oncologist pursue some form of treatment. So it's around 50% across cancers.
Perfect.
And just to comment on the costs. So the costs are high, but pet owners are motivated to pursue the best for their animals, and so they do spend a lot for the conventions of care. All of the trials that we do are fully funded, so the pet owners don't have to pay, and that becomes an incentive for them to participate in these clinical trials.
Perfect. So one of the other questions that came up had to actually do with drug dosing. So can someone comment on the translational ability of drug dosing going from dogs to people? And how do we do that on the veterinary side?
Yeah, so the simple answer, there's no formula. There's a clinical trial that needs to be done. So you take the human drug, you give it to a group of dogs in a 3x3 or other conventional tolerability-like study that we've done in humans, and you assess what dose is tolerable, you assess the pharmacokinetics, and then you give that dose to dogs more chronically. And that's actually a mistake we've made in translating drugs into osteosarcoma metastasis settings where you need to give the drugs for long periods of time. We've done all the studies with rapamycin, for example, in short periods of time and assumed we'd be okay over the long period of time, and that didn't turn out to be true from the standpoint of tolerability. And that's probably why we need to come up with a better version of the formulation of rapamycin.
I'll also comment on the dosing question. So I think that especially if you're using immuno-oncology platforms, I think that while the canine immune system is very similar to the human immune system, especially, say, compared to a mouse, there are important differences that you don't really know until you translate it. So, for example, the human dose that I think is going to wind up being the dose that we're choosing as the maximum tolerated dose in patients with brain tumors is actually 1/100th of the dose that we were giving to canines. So thankfully, we started at a very low dose, and we actually didn't expect it to be biologically active when we moved into humans. But it wound up being that, no, actually it was much more active in humans than it was in dogs. So as you're translating it, I think that you should have some good people at the FDA who will help you think through these things, but you may be surprised at kind of the difference in dosing that you need to achieve for immune-based agents.
I'll add one more thing too. I think it's important when you're doing a lot of your preclinical benchtop work, even in cell models or especially mice, is to look in the literature, see if there's PK that's been done in humans, or even that sometimes the dog data is published, and make sure you're using clinically achievable or pharmacologically relevant doses, so that if you're seeing a signal in those preclinical models, that you have a high probability of achieving that, even going from your mouse to your dog study there. All too often, sometimes you'll see really high doses of drugs used in those studies that you'll never get even in a mouse.
We have a question in the back here.
Perfect.
Thanks, great presentations. This is Brian Ladle from Johns Hopkins. Maybe a question for Nikki and Amy as far as the precinct or those efforts. So I'm curious, for instance, if I'm going to do an immunotherapy in humans, I'm absolutely, being a T-cell centric person, right? I expect like checkpoint inhibitors are going to be required for that to work optimally well. I guess I'm surprised, for instance, in canines, right, where do we stand as far as being able to have those reagents that you can test in dogs that then would give us a maybe even a better launch point to human clinical trials? Maybe it's a discussion for the next 2 hours as well. But, yeah.
So there are several groups that are developing canine checkpoint inhibitors. Some of them are fully canine, some are chimeric. So I would expect to see that single agent data coming through clinical trials, and some of that's actually already been done. Merck Animal Health has a PD-1, I think. And NCI is developing a canine CTLA-4 agent, and as well as others that are in the pipeline. So we'll be doing some work. That's our COTC-031 trial. So yeah, I think that data is coming. I think the issues have just been with creating those molecules and dealing with just the manufacturing and scale and things like that. Do you have other comments?
Yeah, I think people are starting to focus on it. I can give a plug. We applied for a small business grant that actually came out from NCI for that very purpose, to basically develop these sort of biologics that could be used as tools for the comparative oncology approach. So, we have a fully canine phage display platform that we can utilize to try to rapidly generate these. And again, there's efforts, obviously, at NCI and so forth. So, I think you can contact Amy, you can contact myself, I can put you in touch with NCI or with other people who are developing those. So watch this space, I think probably is the thing to say.
Thank you. We have 2 more questions, one here in the center.
Yeah, so I have 2 quick questions, one to John specifically. So you showed that you do radiography on the lungs of your dogs, right? So I'm wondering, do you have an equipment at your center to do PET-CT? Because I think PET-CT for lung nodules is much more sensitive and would show you really, if not micromets, but really, really tiny lesions which might not be seeable on radiography.
That's a really great question. Unfortunately, we don't have the ability to do PET-CT at our institution. It is something that I think we've been trying to develop, but I completely agree. If we could get PET-CT, I think that would be really useful. And actually, it's something that I'm hopeful to potentially incorporate into the human clinical trial if we can get some funding for it.
Thank you so much. And my second question is to the whole panel. So in human patients, like the overall survival, I guess, is the most important thing. So in dogs, taking into consideration that many of them get cancer, like maybe when they're middle-aged in dog years or even old, is it possible to show the age-corrected overall survival for whatever clinical trial is ongoing, or is it very challenging?
I'll take that one. I think survival in general in veterinary studies is problematic. So we tend to focus more on progression-free survival or disease-free interval or those sorts of measures of disease response, because survival is just so fraught with the complicating issues of euthanasia. That's just my 2 cents.
Yeah, I would agree with that. I would just add that age-corrected gets confounded in dogs by their size.
Yeah.
So old, large breed dogs live shorter periods of time than small breed dogs, so it's very complicated to try to correct.
And our last question.
Perfect.
I just wanted to add to the biologics question. So a challenge and an opportunity is not only that you have to make the molecule, if you want to translate it to humans, it has to have similar characterizations for PK/PD, target engagement, and potency. And the challenge, especially with the target engagement and potency, is those assays differ from company to institution. And so if you're not using the same assay that was used to develop the human drug, it's really hard to say that you're going to have the same potency. So it would be nice if pharma would partner a little closer and kind of make sure those canonized molecules have similar characterization to what they did internally.
Yeah, I can speak to the CTLA-4 development path. NCI, not me, extramural NCI, developed companion PK/PD assays alongside the development of the molecule. So, of course, it was going to be important for us to understand the implications of exposure. And so those assays had to be purpose-built alongside the molecule itself. So I totally agree.
Perfect. Well, I think we're out of time. I don't want to cut into the next session. There are a few other good questions on Slido, so if people want to come up afterwards, please do so. But let's give a big hand to our panelists for a great discussion. Thank you.